Sounds like something. All right, chapter 3. It takes so many buttons to get the slides up over here. You can do it. All right, chapter 3. So we've talked about basic behavior of atoms and chemical bonds, right? So now we can start looking at specifically molecules and compounds that are important in biological systems. So that's where we're going. That's where we're finishing up this unit. Biological molecules. So generally, biological molecules are kind of all centered around carbon and the behavior of carbon, right? We are carbon-based life forms. The behavior of the element carbon is kind of crucial in how basically everything inside of you works, right? So we're going to talk about carbon, how it behaves, how it forms compounds, right? We're going to get a bit more into the chemistry weeds just for a second. And then we're going to actually walk through the different biological macromolecules, which will be things that you've probably heard of, at least like kind of casually before, right? These are lipids, proteins, nucleic acids, which is your DNA, and carbohydrates, right? So we'll walk through those. But first, carbon. Carbon, it's right up there. It's right, it's kind of towards the right, not as bad as oxygen. It's kind of in the middle-ish, really. It's got 4 valence electrons, right? Which is kind of a key component of why it is so critical in biological molecules. It's like the ultimate tinker toy. It can form as many bonds, like it can form a lot of bonds. They can go a lot of directions, right? We always draw them flat, but that's because I'm drawing in 2 dimensions, right? It can form chains, it can form branches, they can make rings, balls, tubes, coils, right? You can make molecules in like basically almost infinite number of shapes, right? So this kind of diverse behavior of carbon allows us to make a whole diverse set of stuff, right? I'm just shaking around a bunch of carbon molecules, which is a really weird, abstract way to think about your body, but that's what you are, right? So carbon. Now, we briefly mentioned organic molecules, right? These are molecules with these carbon-hydrogen bonds. Some molecules only contain carbon and hydrogen, and they are, quite logically, called hydrocarbons. So a molecule that only contains carbon and hydrogen is a hydrocarbon. Now, CH bonds, carbon bonded to hydrogen, those chemical bonds, those covalent bonds, store energy. Right? The oxidization, the breaking of those bonds releases energy. So because we can store energy in carbon-hydrogen bonds, they make good fuels. They're good for storing energy, right? So these are your sugars, your carbohydrates, your fats, gases, propane, petroleum, those sorts of things. They're all made up of these carbon-hydrogen bonds because they're good for storing energy, right? All right, hydrocarbons. So you've got this carbon, and this is where carbon is super handy. Since it has 4 valence electrons, we can stick 4 hydrogen on there, right? We can hold 4 bonds in this one molecule. Now we can keep doing this. We can chain a bunch of these together. Right? You ever play with Tinker Toys? You can just keep sticking these together. They make good fuels. This is propane, C3H8. It's a hydrocarbon. It's only carbon and hydrogen. And so it's these 3 carbons. They're holding 8 hydrogen bonds. So it's just storing energy. All of those bonds are holding energy. Ultimately, the length of this carbon chain is potentially unlimited. And so a lot of these biological molecules that we're going to talk about, they're huge, molecularly speaking. Huge carbon chains. All right, so side note, thinking about chapter 2 again, carbon and hydrogen have similar electronegativity. So a hydrocarbon is what? Or hydrocarbons are what? How could we describe them? So water is made of oxygen and hydrogen. They have very different electronegativities, and water is What's the adjective? What's the word? It is a what molecule? A what compound? A what covalent bond? Water is, it's got partial negative and positive charges because the electronegativity is unequal, and so it is
hydrophobic.
It's not hydrophobic. That was in the same. So, I mean, well, water is very hydrophilic, right? Because it's cohesive property of water. But it is hydrophilic. It is cohesive and adhesive because water molecules are, starts with a P. Polar. Yeah, polar. All right. All the way back to this little thing. Carbon and hydrogen have similar electronegativity, so hydrocarbons are non-polar, right? And so they're non-polar. They don't have partial charges, so are they hydrophobic or hydrophilic then? Back to hydro. The other one. Wait, okay, now I'm just having like a Vincent mask problem. All right. They don't have partial charges. They are not polar. They are non-polar, so they are hydrophobic. They're hydrophobic. All right. All right. So hydrocarbons are hydrophobic. They are non-polar. That will be important at the end of this chapter. All right. But most of the molecules we're going to talk about are not just hydrocarbons. They're going to be a lot of carbon and hydrogen and other stuff. All right, so if we've got a hydrocarbon and we attach other stuff, the other stuff is called a functional group. It's a group that's going to add function to the molecule, right? So we've got our carbon-hydrogen core or carbon-hydrogen skeleton, however you want to say it, right? This kind of baseline hydrocarbon molecule. And we're going to start sticking different stuff on it, right? When we stick different things on it, we will change the electronegativity. Yeah, for example, a hydroxyl, an oxygen hydrogen, or a carbonyl, C double bonded to O, right? And you can always, when we start adding oxygen, right, because oxygen is way over here to the right, it's very electronegative. We're going to start, right, oxygen's greedy. We're going to become electronegative. We're going to create positive, negative, partial negative charges around these oxygens. Now, we are creating regions of these giant molecules that are polar. Right? For our purposes, I'm not making this chain 100 carbons long, but you can imagine, right? We've got all of this hydrocarbon. It's all non-polar. It's all just kind of hanging out and chill. But there's an oxygen over here. It causes a partial negative charge. Now, this end of the molecule is going to behave differently because we've added kind of a partial negative charge to one side. All right? They become polar because of the electronegativity of oxygen. Now, there are loads of functional groups. They do all kinds of things. We're not going to memorize huge, enormous lists of functional groups, but if you want to read more about them, there's a table in your book, right? And this is where we start incorporating all those other elements we see in biological systems, right? Phosphorus, sulfur, nitrogen. Where was another one? Well, there's more hydrogens. Lots of oxygen. There's always lots of oxygen around, right? And they can be basic, they can be acidic, they can be polar, they can be hydrophobic, right? And so they're just going to kind of start imparting all kinds of functions onto these organic molecules. You don't need to memorize this table. It's just there for, you know, for you to see. All right. Cool. We've got hydrocarbons. Got these organic molecules. We can stick stuff to the molecules so that they can then do things. We'll talk about the things they do later. Now, when we start sticking things onto these hydrocarbons, we get another way kind of talk about and describe these molecules. We start to create isomers. All right, so isomers are molecules, organic molecules, with the same molecular formula, but different structures, right? So we've got two molecules right here. Each one has 3 carbons, 8 hydrogens, and an oxygen, right? 3 carbons, 8 hydrogens, and an oxygen. They're made of the same numbers of atoms of different elements, right? The same pieces are in each, but the structure is different, right? In this carbon core, this kind of carbon skeleton, right? Frequently, just to kind of make things simple, we'll number these so we can talk about them. So let's say we've got carbon 1, carbon 2, carbon 3, right? Carbon 1, carbon 2, carbon 3. So we've got the same number of all our different kinds of atoms here, but in this molecule, the OH group is on carbon 3. And this one, the OH group is on carbon 2. So the same pieces are there, but they're in different places, right? Specifically, in this case, they're attached to different carbons, right? Here are my hydroxyls on carbon 3. Here are my hydroxyls on carbon 2. So that is a structural isomer. The carbon skeleton structure is different, right? We've attached different groups to different carbons. Now, we can also have something called a stereoisomer. In a stereoisomer, the carbon skeleton is the same, right? We've got carbon 1, 2, 3, carbon 1, 2, and 3. So carbon 1s both have 3 hydrogen, carbons 2s both have 2 hydrogen, and carbon 3 double bonded O and a hydrogen, carbon 3 double bonded O and a hydrogen. So the pieces are attached to the same carbons, but because carbon, right, has kind of these 4 directions that it can bond to things, you can kind of spin things around, right? So here we've got our oxygen oriented in this direction, and here the oxygen is oriented kind of straight out from the molecule, right? And because oxygen's in a different place, it moves the electronegativity, the molecule is going to behave differently, right? But the oxygen is attached to the same carbon. So the functional groups are arranged differently in space. So sometimes these are called spatial isomers. We've not changed what carbon it's on, we've just kind of spun it around in space, right? Same skeleton, we just have stuff kind of pointing different directions, right? That's a stereoisomer. Now, we can also have, because carbon makes 4 bonds, a special case of a stereoisomer called an enantiomer. Enantiomer. In an enantiomer, right? It's a stereoisomer, so we've kind of changed where things were. If you think about kind of the directions these are pointed here, like if you pull the OH to the front, here the carbon H3 is pointed kind of to the left, here it's pointed to the right. So they're arranged differently in space, but they are mirror images of each other, okay? Molecules that have mirror images are called chiral, right? So this is where that right here, we've got the CH3 on the left, CH3 on the right, where you get left-handed and right-handed isomers. Sometimes they talk about it that way in chemistry classes. I never did amazingly in chemistry classes, but it's a thing, right? So enantiomers are stereoisomers that are mirror images of each other. It happens with carbons because of this kind of arrangement of 4 bonds.
So why would they be mirrored? Like, what's an instance where the molecules would need to be mirrored at all, if that makes any sense?
Okay, can you say, oh my gosh, it is so loud in here. Can you say that one more time, Lakin?
Why would the molecules need to be mirrored? Like, what's the, can you give me an example of something that, the reason why they'd be mirrored, or what causes them to be mirrored, besides that it's just...
So I wouldn't say that anything causes them to be mirrored, right, in kind of like an active reasoning sort of way. It's just atoms and molecules come together given the behavior of their electrons. And so you will end up with, right, these covalent bonds forming, and when different molecules form, they may simply form slightly differently, right? So there's not like some other force that is like making left-handed molecules and making right-handed molecules necessarily. It's just when we observe molecules, there's usually some mix, right? If you look at some kind of batch of this substance, you will find some of these molecules that are existing in their left-handed form and some that are existing in their right-handed form. So it's more of like an observation and description of how these things exist than like any kind of like process, if that makes sense. Does that make sense? Did I make it worse or did I make it better?
Yeah, that made sense. So
Okay.
does that mean since they're flipped and they're mirrored, would that make the oxygen-hydrogen molecules connect, or would they still repel each other?
So in this case, right, the hydroxide stays the same, right? Like it stays associated. The spatial change is kind of between this CH3 and this hydrogen atom. So it's hard to do with like, I should have just like stolen some models from the lab. When you're in lab, you'll see like these ball and stick models where you can like take them and turn them. But right, so basically what's happened is this is like carbon bonding, like we're looking at these bonds 1, 2, 3, and 4. So on carbon's valence electron 2, it's a hydrogen. And on this one, it's where they put the CH3 and then they put the hydrogen on bond 3, basically. So it's just kind of.
It's kind of like you have like your grid 1, 2, 3.
Okay, thank you.
Yeah. Yeah. And again, this is much easier when you have like a physical model. Is everybody, if you guys are in the lab, you'll do this. You'll be putting molecules together and actually see how the bonds work. It's a lot easier than trying to wrap your head around it in 2D. So right, so enantiomers, special case of stereoisomers where your isomers are mirror images of each other. All right, so behavior of carbon, check. Now let's start building some big biological molecules. So for the rest of chapter 3, we're going to be working through these 4 groups. We're going to get into carbohydrates today. They're a big group, so they'll take a bit of time. And then Monday and Wednesday, we'll wrap up DNA, proteins, and lipids. Not right this second. We're going to go through them each in turn. So, but first, I keep saying macromolecule. What the heck is a macromolecule? It's a big molecule. Easy enough. More specifically, we're going to be talking about polymers. Okay, and a polymer is just a large molecule basically made up of repeating pieces. Right, so a polymer is a big molecule made up of a long, basically string of monomers. Okay. And of our 4 groups, they are all polymers made of monomers except lipids. And we'll talk about why lipids, what is different about lipids when we get to them. All right. Now, the nature of a polymer, the way a polymer behaves, depends on the monomers that it's made of. That makes sense. Right, so starch, which is a complex carbohydrate, a polymer, is made of monomers that are just simple ring-shaped sugars. Right. DNA is made of nucleotides, right? Long strings of nucleotides that cause DNA to coil and have its DNA shape, but that also carry information and give it its function. Right, so when we kind of talk about each of these molecules, we're going to talk about their monomers, how that affects their structure, and then how that structure then gives them their function. Okay, so polymers made of monomers, right? These polymers are formed and broken by 2 specific kinds of chemical reactions. Right, so these polymers are these long chains of monomers. These monomers are connected to each other through dehydration reactions. So one monomer is attached to another monomer in a dehydration reaction. Subsequently, when polymers are broken down, right, one monomer is broken off of another monomer by a hydrolysis reaction. if we had to guess, we're going to put monomers together by taking something out. So if it's a dehydration reaction, what are we probably taking out to hook these monomers together? If we're dehydrating them, what are we losing? What do you lose when you dehydrate stuff? Water, right? That's exactly what's going to happen. So in a dehydration reaction, we've got these two monomers. One of them is going to lose an OH. The other one's going to lose an H, right? H, H, and O, like H2O, that's water. It's lost. And the two spaces where the hydrogen and hydroxide were then form a covalent bond between the two monomers. In hydrolysis, lysis is just breaking, right? So we're going to break it with water. So same thing, water is added in reverse, right? We're going to add water to a polymer and break the monomers apart. So for example, dehydration. We've got two monomers that are just made up right here, right? Two little 3-carbon molecules. We're going to put them together in a dehydration reaction. So we've got a hydroxyl and a hydrogen there in the middle. Those are going to pop off and be lost as water, right? And so when we take those off, we've got an open space right here, right? On either side where we took them off. There. And so to make these big polymers, that's what's happening, right? All down, these little monomers are being added on. We're removing water and sticking another monomer on, removing water, sticking another little monomer on there. We make these giant polymer chains. Subsequently, if you want to metabolize something, right, we eat sugars and then we want to break them apart to use the energy in them. They're broken apart in hydrolysis reactions, which is just the exact same thing in the other direction. We put an H and an OH in there and we break them back apart. So your body can build polymers, it can break up polymers, and it's happening all the time in your cells. All right. Sometimes, and depending on who's teaching your lab, if you're in the lab, so these dehydration reactions are sometimes called condensation reactions. It kind of makes sense, right? When things condense, right? That's like water coming out of the air. So you can kind of relate that to forming polymers where we're pulling water out of the monomers, right? And frequently in biological systems, there are enzymes that are helping with this. When we talked about chemical reactions and reaction rates and that we can speed them up using enzymes, this is one of those times where that is happening. You have enzymes in your body that are working to make and break polymers, depending on what your body's needing to do, right? And that process is called catalysis, right? Positioning and breaking the bonds between monomers and polymers. All right, I think this gets us into, yep. So macromolecule group number 1, carbohydrates. Everybody loves carbs, I feel like, if we're honest, right? Everybody loves carbs. So we're going to talk about the structure of carbs, of sugars, of complex carbohydrates, and then talk about these polysaccharides, right? These polymers of sugars and their functions in our body. Well, we'll start it today. We'll finish it on Monday. So carbohydrates, right there in the name, carbons and hydrogens, 8s. Carbohydrates contain carbon, hydrogen, and oxygen, specific ratio. So they have, for every 1 carbon and 1 oxygen, they've got 2 hydrogen, right? So that molecular formula then is CH2O, right? Basically what this little guy is right here. This is a CH2O. And so it's lots and lots and lots and lots of these all connected together. So we tend to kind of write it out at CH2ON, right? So if I had like 12 of these all tacked together, it becomes CH2O12, right? So it's full of carbon-hydrogen bonds. So carbohydrates are good for storing energy. Lots of carbon-hydrogen bonds. lots of stored energy in a carbohydrate, right? So they release energy when they're oxidized, when they lose electrons, and so they're good for energy storage. Or just for being stored, depending on how many of them you consume. Right? So our monomers, right, our smallest pieces in carbohydrates are called monosaccharides, right? Saccharide is just sugar, mono is one. So our one little piece of sugar, molecularly speaking, not like a sugar crystal, right, is a monosaccharide. They tend to have 3 or 6 carbons. We're going to talk mostly about 6 carbon sugars because they're the ones we hear about most often, right? And so 6 carbon sugars, so it's CH2O, 6 will be our molecular formula, right? We've got 6 of these little carbon bits. If you do the math out, right, you've got C6H12O6, 6 carbon sugar. All right. They can form chains. That makes sense, right? We've already talked about carbon. Everybody loves a carbon chain. We can make a carbon chain with 6 carbons in the middle and then 12 hydrogens and 6 oxygens dispersed around the outside in some fashion. Now. Glucose. We've heard of glucose. We've got 6 carbons. We've got different hydrogen and hydroxyl groups tacked onto each one, right? Like we would expect. But, of course, these atoms, these molecules can come together in different ways. So if we have the same molecular formula and they're coming together in different ways, then we are making what's? Same formula, different arrangement is an, starts with an I. Isomers. Isomers. We're making isomers. Yeah, you got it, right? So we have glucose, fructose, and galactose, 3 common 6 carbon sugars in us, right? Glucose has 6 carbons, and we'll call this one carbon 1 up here just to make our life easy. So carbon 1 in glucose, right? Double bonded oxygen, hydrogen. Carbon 1 in fructose, something completely different. Carbon 2 has a double bonded oxygen. Carbon 2 here has an H and a hydroxyl. What kind of isomers are fructose and glucose? It's on the screen. It's labeled. It's small, but yeah. It's a structure. It's a structural isomer, right? Because the carbon skeleton is different. Carbon 1 is double bonded to an oxygen in glucose. Carbon 2 is double bonded to an oxygen in fructose. The groups are on different carbons. Structural isomer. Galactose, all right, carbon 1, yeah, sure, keep going. Womp, what happened? Yeah, same carbon, different direction. What kind of isomer? Stereoisomer. Same carbon skeleton. Things are pointed in different directions.
Does that make
So they behave
glucose just a
differently.
normal isomer?
Say again?
Does that make glucose just a normal isomer?
So, well, it's all relative, right? Because you could also compare fructose and galactose. And you would have, right, fructose is a structural isomer of galactose. So we're just comparing it to glucose because that's what I put in the middle. So it's all kind of relative to the molecule you're looking at. So glucose is a stereoisomer of galactose, but it's a structural isomer of fructose. Okay, let's see if we can. All right, it can form chains, but those chains dissolve in water and turn into rings. So in your body, these sugars usually exist as rings. All right, so we can still see these, right? We've got glucose and fructose, right? Our structural isomers, right? We've got fructose is weird. It's got like carbon, are we numbered? Yeah. 1 out here in space, 2, 3, 4, 5, 6. Versus, you don't have to, you're not memorizing this. Don't memorize this. Well, maybe for lab, but not for me, right? Carbon 1 is actually in a 6 carbon ring. Carbon 6 is hanging out there. right? So basically, we get isomers again. This time they're in the form of rings. There's galactose, glucose. Our stereoisomers, so they look more similar as a ring. All right, now, real fast, we can get through disaccharides. So disaccharides are where we take two of our monosaccharides, two rings, and stick them together, right? Two monosaccharides makes one disaccharide. And this is a transport form of sugar. So we eat glucose, our body metabolizes glucose for energy. But that means that your body is full of enzymes made to break down glucose. So glucose can't travel alone. It'll get et, right? So if your body wants to move glucose, it has to do something to protect it from enzymes that will break it down. So it attaches it to one of these other isomers, one of these other monosaccharides, so that it can move around. When it gets where it's going, we break it back into monomers and it can be metabolized. So examples, glucose attached to fructose makes sucrose, that's table sugar. Glucose attached to a galactose makes lactose, that's milk sugar. Yep. And right, wherever this gets when it's going, when it gets where it's going, right, it's got to be broken down. Lactase is the enzyme that breaks down lactose so that you can then metabolize this glucose, right? In adults, the levels of the enzyme lactase goes down. You can't break down lactose. You get all full of lactose. That's how you end up lactose intolerant. Yeah. Yeah, and my husband, we just thought he was like randomly like unwell in his gut, just as a normal condition. Boy just needed some lactate. That was it, right? So as adults, we frequently become to some degree lactose intolerant because we produce less lactase, which you need to get the galactose off your glucose so you can metabolize it. That's a whole, no, that's another other bag of chips. Will we get into gluten? I don't know. I'll think about gluten. I always think about gluten. All right. Good, perfect. That gets us to polysaccharides, right? The big complex carbohydrates, which we will talk about on Monday. Perfect. I like it. So quiz and lecture journal are open. Don't forget about them. Be working on your study guide. Don't forget you've got an exam a week from today. If you need anything, I'm right over there pretty much all the time. What? Monday. So lecture journal and quiz are both due by like 11:59 Monday night. So you've got like the weekend. Yeah, thanks, you.